Welcome everyone to this week's edition of the Extracellular Vesicle Club. Today we are going to do something a little bit different from our normal format, which is when authors of a paper present their own work and then we discuss that with them. We're actually going to have some original research here, and we're going to go over no fewer than 3 papers that have appeared in the last few years in the literature, and specifically on the subject of neutrophils and EVs. I actually get a lot of questions recently about neutrophils and how they're interacting with extracellular vesicles. So hopefully we'll learn a little bit about that today. And before I start, before I introduce our guest today, I would just like to remind everybody to put your comments and your questions in the chat box, and we will allow unmuting at the end of the presentation. I also just want to put in a very quick plug for an ISEB meeting that's coming up, which is EVs in nervous systems. Central nervous system, peripheral nervous system, systems of a lot of different organisms. This is going to be a meeting that takes place in Rome, Italy, from the 6th to the 8th of December, 2023. So please look that up on the ISEB website and get your abstracts and registrations in. So today, as I said, we're going to talk about neutrophils and EVs, and I'd like to introduce you to Anne Ramsey, who's a very hardworking and insightful scientist who's been working in my group this summer. And part of her summer project was to prepare this presentation to help educate us on neutrophils and EVs. So Anne, I'd like to ask you, first of all, if you could tell us a little bit about your background and what you are doing at Johns Hopkins this summer.
Yeah, so I am a veterinary student from Oregon State University's Carlson College of Veterinary Medicine. I'm currently between my first and second years and have been enjoying my time there. This summer, I am at an NIH-funded fellowship at Johns Hopkins. It's the Veterinary Research Scholars Program. So this program is hosted at a number of universities, and I was very lucky to be able to do it at Hopkins. It allows us to go to the National Veterinary Scholar Symposium, which I attended a few weeks ago.
Great. Well, and thank you for agreeing to share with us today. I know that EVs in veterinary medicine it's a big topic. It's one that's increasing, and we rely for a lot of our knowledge of EVs on animal models, lots of different, diverse animal models that are out there, and that can help us to understand human health. And in fact, we have this term One Health that applies to the health of a lot of different organisms. And how our relationships with each other, our health is really interconnected. So Anne, I'd like to invite you to go ahead and share your screen, and we look forward to hearing about these papers. I'm going to be looking through the participant list. We've invited some of the authors also to join, so just in case they're here, please feel free to reach out to me, and we can also allow you to help out with the discussion. That'd be incredible. Go ahead, Anne.
Awesome. Thank you so much. So before I start my presentation, I just want to do a little brief review about EVs in veterinary medicine. This is a really interesting paper that's looked at EVs in vet med research over the last few years. So there has been increasing interest in EVs in vet med, especially since like 2017, 2018. They've looked at a variety of applications, most predominantly physiology and pathophysiology, but there's also been some biomarker and therapeutic work. A majority of these papers have looked at domestic animals, especially in cows, but there have been some papers in exotic and wild species as well, which is interesting. And then most of these EVs are derived from milk, but there's also been a lot of EV work with cell culture and reproductive fluid in animals. So this is not the main focus of my presentation, but just to highlight, One Health is a really important concept in the interconnection between human and veterinary medicine when it comes to EVs. So to begin, I looked at the association between extracellular vesicles and peripheral blood neutrophils this summer. I'm here today to address what we know about neutrophils and EVs. We know that neutrophils release their own EVs, and these may have therapeutic applications. However, there are actually very few papers out there that address neutrophil association or binding to EVs. Therefore, the main question that I've asked this summer is, do EVs interact with neutrophils, and if so, how? I want to know how exogenous EVs administered to an organism for any purpose impact or are impacted by that organism's immune system. Neutrophils are the most abundant white blood cell type in humans, comprising over 60% of leukocytes. And these are considered the first responders during acute inflammation. In this presentation, I'll provide some background on the significance of neutrophils in the immune system, and then review several papers that look at EV-neutrophil interaction. Finally, I'll discuss some recent data from papers from the Witwer lab. Now, keep in mind that I'll only be discussing a handful of studies, and my background in EV research is limited to the last few weeks. So if you're involved in any research with EVs and neutrophils or know of any other recent studies, I invite you to share your knowledge during the discussion portion after my presentation. My lab primarily focuses on EV interaction with peripheral blood mononuclear cells, or PBMCs. These are distinguished by their round nuclei in comparison to granulocytes, which have multi-lobed nuclei. PBMCs are composed of lymphocytes and monocytes, whereas granulocytes are composed of neutrophils, eosinophils, and basophils, and neutrophils are by far the most common granulocyte. These all have a variety of functions in innate and adaptive immunity. PBMCs and granulocytes all originate from hematopoietic stem cells in the bone marrow. This differentiates into myeloid progenitor cells or lymphoid progenitor cells, and these may be distinguished by where they mature. So lymphoid progenitor cells typically mature outside of the bone marrow, whereas myeloid mature within the bone marrow. These then give rise to our granulocytes as well as our PBMC cell populations. Neutrophils are actually a heterogeneous cell population. Their phenotypes differ based on the stage of their life cycle, known as aging, or their microenvironment. After density gradient separation, most neutrophils will be present in the granulocyte layer, but some may also contaminate the PBMC layer in certain inflammatory conditions. The image in the upper right here depicts neutrophilia, or excess circulating neutrophils in the blood, in which you may appreciate the multi-lobed nuclei of neutrophils. They have a half-life reported between 6 and 12 hours and may be produced in the bone marrow up to 10 to the 11th cells per day. The general life cycle of a neutrophil involves differentiation and maturation in the bone marrow, followed by release into the blood, and then migration and recruitment to specific tissues, and finally clearance. The different maturation stages of neutrophils are depicted on the right here. There are many mechanisms regulating neutrophil retention in the bone marrow and released into the blood that vary between homeostatic and inflammatory states. One key mechanism is the interaction between two chemokine receptors, that's CXCR4 expressed on immature neutrophils and CXCL12 expressed on osteoblasts and bone marrow stromal cells. This interaction is disrupted by GCSF, or granulocyte colony stimulating factor. GCSF also stimulates differentiation of granulocytes and is modulated by IL-17, released by T lymphocytes. And this is also regulated by IL-23, released from macrophages. This diagram depicts neutrophil recruitment from the bloodstream. This mechanism may vary depending on the organ and inflammatory state, but generally speaking, the process involves neutrophil rolling, adhesion, crawling, and transmigration. The process begins when leukocytes encounter a pathogen in the tissue and they release inflammatory mediators like cytokines that alter the surface of endothelial cells. This upregulates selectin proteins on the endothelium, which bind free neutrophils, allowing integrin to adhere neutrophils to the endothelial surface. There, neutrophils encounter chemokines, which induce paracellular or transcellular migration from the vasculature. Neutrophils then migrate to sites of inflammation in response to a series of chemoattractants released sequentially or hierarchically for guidance through the tissues. The main function of neutrophils is to kill pathogens. They do this in a variety of ways, including releasing cytoplasmic granules composed of proteinases and other destructive molecules. They can engulf microbes through phagocytosis, in which they release their granules and reactive oxygen species, or ROS, into their phagocytic vacuole. To destroy microbes under shear conditions in the blood, neutrophils eject their own chromatin covered in toxic molecules like elastases. The release of this chromatin is known as netosis, or the release of neutrophil extracellular traps. Neutrophils may or may not survive netosis depending on the origin of the chromatin they release. Neutrophils may also modulate the adaptive immune system. For example, they produce cytokines BAFF and APRIL, which are B lymphocyte stimulator and A proliferation-inducing ligand, and these are both critical to B cell function. They can also act as antigen-presenting cells and communicate with T cells. Neutrophils are also known to release their own EVs. This will not be the focus of this talk, but it's a very interesting line of research as well. Circulating neutrophils may be cleared in the mouth, eyes, lungs, or bone marrow. In states of inflammation, apoptotic neutrophils are cleared by macrophages. This then inhibits macrophage release of IL-23 and contributes to the regulation of neutrophil release from the bone marrow in general. So I'll now review several articles that analyze neutrophil association with EVs from 3 very different EV sources. Please note that the EV separation and labeling protocols differ between these studies, as well as the EVs themselves. So I encourage you to interpret these results in the context of their methods. In 2021, Santos Coquia and colleagues characterized goat milk EVs and evaluated EV-neutrophil interaction in a peritonitis mouse model. They labeled EVs with sulfocyanine 5, which is a protein marker, and administered these EVs to mice with peritonitis as well as control mice. So the general study design involves creating a mouse peritonitis model and then injecting these mice with EVs through the tail vein and collecting peritoneal fluid for analysis through flow cytometry and confocal microscopy. This figure confirms the association between labeled EVs in both neutrophils and macrophages. Figure A depicts the gating strategies for the control and EV-treated group. So controls are up top here, EV-treated groups are on the bottom, neutrophils on the left, macrophages on the right. Figures B and C depict association between EVs and neutrophils, measured through mean fluorescence intensity up top and then percent positive population on the bottom. The cell groups are diagonal from each other, so neutrophils are here in the upper right and the lower left. It's interesting to note that macrophages tended to have a greater percent positive population for association with EVs by about 10% compared to neutrophils. These confocal microscopy images show the various markers used to differentiate EVs as well as the different cell types. So on the far left, this is their EV signal in white. We have neutrophils in red and then macrophages in green. And the merged image on the far right uses a blue marker for the nuclei. But the white arrow indicates EV fluorescence within the perinuclear area of the neutrophil in what authors describe as rounded packages. And then, of course, down here we have our macrophages. So moving on to our next study, Cermiak et al. And colleagues harvested EVs from patients with granulomatosis with polyangiitis, or GPA, in 2020. GPA is an autoimmune disease of unknown etiology that involves anti-neutrophil cytoplasmic antibodies and vascular inflammation. They investigated the interaction between EVs and neutrophils by comparing EVs derived from platelet-poor plasma in healthy and diseased individuals. They used PKH67, which is a fluorescent tracker dye that is of lipid nature. And that was their main marker for flow cytometry. So this group separated neutrophils from the peripheral blood of healthy patients, then incubated neutrophils with EVs with or without first incubating them with GM-CSF to prime them. These EVs were either derived from GPA or healthy control patients, and then they did several assays such as EV-neutrophil interaction, ROS production, netosis, and then a targeted oxylipin analysis of methods specific to those assays. So in this figure, they demonstrated that only EVs released from the GPA patients bound human neutrophils that were first primed with GM-CSF. So in this figure, the y-axis shows the PKH67 positive cells, the cells positive for that EV marker. And then we have our EVs derived from GPA or disease patients, and then HC healthy control patients. And so again, the only significance they found was in those EVs from GPA patients when they were incubated with neutrophils, and the neutrophils were first primed with GM-CSF, so not just neutrophils alone. But they did find a positive association here. So in these figures, they looked at ROS production and double-stranded DNA release, so that's your netosis, using C5a as a positive control in this blue color. Again, they found that only these EVs from GPA patients with the activated or primed human neutrophils had any significance. There was increased ROS production and DNA release for this treatment specifically. So this group followed up with a targeted oxylipin analysis and demonstrated that LTB4 and 5-oxo-ETE are found in EV-GPAs at significantly higher levels than healthy controls, and these may activate neutrophils in a pure chemical form. So these graphs just demonstrate the pure chemicals being incubated with neutrophils and resulting in ROS production and double-stranded DNA release. Moving on to our final study here, in 2022, Diaz-Godinez and colleagues investigated the interaction between human neutrophils and EVs derived from the protozoan parasite E. histolytica. This parasite may be ingested from contaminated water or food sources and induces an inflammatory response involving neutrophils. This group examined the activation of neutrophils by 3 EV types. EVs derived from the parasite at the trophozoite stage, from neutrophils themselves, and then from a trophozoite neutrophil co-culture. I will mostly be focusing on the EVs derived from the parasite. They centrifuged these 3 culture groups and collected the supernatant for EV harvest. So for the interaction assays, parasite-derived EVs were stained with DIL, which is a lipid dye, and incubated with human neutrophils separated from peripheral blood from healthy subjects. Cells were then formaldehyde fixed, stained, and examined through confocal microscopy. ROS and net detection assays were performed as well. These were evaluated by incubating fluorescently labeled EVs with neutrophils and measuring fluorescence intensity through confocal again. After EV exposure, neutrophils were stimulated in 3 separate treatments for the ROS production and the netosis. So they used either PMA, ionophore, or trophozoites as some different positive controls for these assays. This figure demonstrates neutrophil interaction with EVs from 3 different sources. So on the left here, we see the sources of the EVs, either the control without EVs, the amoebic derived EVs, which I outlined in a rectangle, and neutrophils and co-culture. And then the images themselves, you're looking at neutrophils under different staining conditions. So most significantly in my pink box at the far right in this merged image, we see EV uptake into neutrophils. Those little red dots are the DIL stained EVs. So they suggest that amoebic EVs fused to the neutrophil membrane and were internalized into the cytosol. In terms of ROS production, unstimulated neutrophils appear to produce a basal level of reactive oxygen species. Stimulating neutrophils with pure PMA, ionophore, or amoeba significantly increased ROS production as expected. They found that parasite-derived or neutrophil-derived EVs apparently decreased ROS production despite the various stimuli. So that is interesting to note. And then this demonstrates our net production in our 3 different conditions, the PMA, ionophore, and then amoeba. So the red that you see here is just the pure chemical for each of the different treatments. And it's interesting to note that the amoebic-derived EVs tended to prolong net release or delay it compared to just the pure chemical, which was our positive control. So we expect increased net production. So these different studies, I'll now kind of compare them all together to summarize. We had 3 very different EV sources, and the EV separation methods also differed. So the first study used a protein marker. The second and third studies used lipid-derived markers. So we know that there is variable promiscuity for some of these markers. So that's something to keep in mind. For EV separation methods, our first study used TFF, ultracentrifugation, and SEC. The second study also used ultracentrifugation. But the third study tended to rely on total exosome isolation reagent. So again, there are some different methods of separation here. All 3 did a fair amount of characterization. They all used the nanoparticle lipid analysis to note. In terms of the source of neutrophils, the first study looked at mice in an in vivo model, whereas the second and third got neutrophils from healthy human blood, and then did an ex vivo model that was primarily, incubating EVs and neutrophils and then adding different markers or stimulants. In terms of results, there was some kind of positive association between EVs and neutrophils in all 3 studies. In the first study that was shown through flow cytometry, again, those are the neutrophils derived from peritoneal fluid. And then confocal microscopy found EVs in the paranuclear area of the neutrophil. In the second study, it's important to remember that they only saw positive association with those EVs derived from diseased patients in neutrophils that were first stimulated with GM-CSF. And in this study specifically, there was positive association, increased ROS, and increased netosis, but only for that one treatment. And then our third study primarily relied on confocal microscopy. They found EVs localized to the cytosol of the neutrophil, a decreased ROS in contrast to the second study, but again, increased netosis when exposed to those amoebic EVs. So the previous studies established that EVs may bind and activate neutrophils under various conditions. I will now address the question of whether exogenous EVs interact with neutrophils through some data from the Witwer lab that I took a look at this summer. Two recent studies from our lab have examined the association between EVs and immune cells in both in vivo and ex vivo non-human primate models. We use EVs harvested from XB293F cells, which are from a human embryonic kidney cell line. And these readily produce EVs and can be engineered for other purposes. So they're an interesting EV source to look at. So in terms of methodology for the in vivo study, EVs were administered to pigtailed macaques, naive specifically, and then blood was collected at different time points after EV exposure. In the ex vivo study, the second study that I will bring up primarily used just naive animals, collected blood from those animals, and then spiked EV into the blood after they were collected. But one note from the first study used an EV exposed animal as well to compare. And then all of these samples were run through flow cytometry in order to determine level of association between EVs and neutrophils. And EVs were engineered to contain palmgret, which is a dual-labeled GFP nanoluciferase fusion protein, and memglow lipid dye was also used for tracking, but I will focus on data from the palmgret dual-label marker. So interestingly, the 2022 study found that after EV administration, over 80% of B cells became GFP positive, indicating association with EVs. About 14.1% of granulocytes, 13.8% of monocytes, and 6.7% of CD3 positive T cells associated with EVs. And remember that granulocytes include neutrophils, so roughly 14% association there. And this was the in vivo study. A blood spike experiment associated with the 2022 study incubated EVs in whole blood from both EV-exposed and naive donor animals at two different EV concentrations. Focusing on the eGFP label, there was greater association between granulocytes and EVs compared to controls. And this was higher at the greater EV concentration. So the more EVs that you spiked into the blood, the greater association with granulocytes we saw, and that was over 25% in both the naive and EV-treated groups. And this graph just shows percent positive granulocytes on the y-axis, and then our control and two different EV concentrations on the x-axis. So the 2023 study out of our lab found similar levels of association between EVs and PBMCs. And remember, this is the ex vivo only study. The greatest association, again, was in B cells as expected. In contrast, there was little association with granulocytes found in this study, which was under 5% at all incubation times, and the incubation times ranged from 5 minutes to 4 hours. So the difference of association between these two studies may be explained by the concentration of EVs spiked into the blood, but this would be interesting to tease out in further research. So in conclusion, to address my initial question of whether neutrophils interact with exogenous EVs, the answer is, of course, yes, no, and maybe. Keep in mind that neutrophils are a heterogeneous population whose activation depends on a complex combination of inflammatory mediators, as well as the disease state of the biological model. There are still many unanswered questions in neutrophil research itself, especially regarding cell subsets within the neutrophil population and cytokine production. When it comes to EVs, there's evidence of neutrophil association with EVs derived from pathogenic and non-pathogenic sources in both healthy and disease states, but this is highly variable. Of course, it depends on if you're doing an in vivo versus ex vivo study, what kinds of EVs you used, how you separated them. So it ends up becoming very complicated. A lot of these studies also tended to prime their neutrophils, which may influence their activation, so that ROS production or netosis. Future research should consider the mechanism behind EV-neutrophil association to better understand how administering EVs to an organism for any purpose may impact the most common type of human white blood cell, the neutrophil. Thank you so much. These are the 3 papers that I used highlighted in blue if you are interested in looking a bit closer at their methods or contacting the authors. Thank you, and let me know if you have any questions.
All right, well, thanks very much, Anne, for sharing all of that information with us. We now have an opportunity to discuss with Anne and with our various neutrophil experts who are on the call any questions or comments that you might have about these or similar studies. So I am not seeing comments yet. Let me just check here because this new format where the chat box is full of people coming and going is a bit confusing to me, but we may have some questions. No, I don't think we have any questions yet. So let me start with a question for you, Anne. The the differences in the methods between these studies, the difference in the separation methods for the EVs. Do you think that reasonably these methods might affect how EVs are interacting with neutrophils?
Yeah, I think it's very possible. And because the methods are so different, we don't necessarily know the purity of the EVs in terms of from a comparison standpoint. So it's difficult to compare methods between different studies that are not exactly the same or do not necessarily adhere to the same um, standards as is typical. So yeah, I absolutely think that because these studies are so different, it's difficult to compare. And it's difficult to know exactly what is going on in this interaction if we are not confident in our EV marker or that what we're looking at are the EVs themselves.
Indeed, indeed. All right, thank you. So, Ming Lin, you have a comment here about the release of EVs from neutrophils.
Yeah, great talk. And we're studying neutrophils and also netosis as well. And actually, in the new PMA-induced neutrophil netosis, netotic cells in the early stage, they also are blabbing EVs. So in the study, in the experiment, when you incubate with exogenous EVs adding to the PMA stimulated neutrophils. So those neutrophils, of course, if you mark the neutrophils you added, then you know which is which. Otherwise, you don't know whether those are EVs you adding there or the total cell generated. Yeah, so just share the experience with you. Thank you.
So Minglin, how do you label your EVs?
Yeah, I actually you're a very interesting question. I did not pay a lot of attention on this interaction yet, but we do find accidentally we found these EVs by when we made the confocal image in the different stages of the netotic cells. We do find those netotic neutrophils, the blabbing EVs, but we did not do a experiment as you guys did, adding the trying to look the EVs interaction with neutrophils. I think you see you have clearly at least several methods to label the EVs. You can try with any of those methods to label the EV, either lipid marker or whatever, then wash it, makes it clean, then add there and look, how do they interact with netotic neutrophils. That shouldn't be very difficult.
Very good. Thanks for the insights, Minglin.
Thank you.
We have a question next from Matt Lawrence. Matt, do you have your microphone there?
Yes. Can you hear me all right?
We can. Go ahead.
This was a really interesting presentation. Thank you for summarizing all this data from the 3 different papers. Was it clear from the papers on how the EVs were being taken up by the neutrophils? Are we looking at phagocytosis, endocytosis, fusion? Did they have any insight into those mechanisms?
Yeah, that's a really great question. I think that is one of the limitations of flow cytometry and confocal microscopy as the main methods that were used, and that we don't have a very good understanding, at least to my knowledge, of the actual mechanism at play. We can look at association, of course, through flow, but that might be simple surface binding or actual internalization. And then the different confocal images show internalization, but again, we don't know if it was through phagocytosis or some other mechanism like membrane fusion. So I think that's a really interesting question that would be great to pursue further with other research.
Yeah, so maybe some future studies could look at some inhibitors of specific pathways to determine whether this is indeed surface association or perhaps one of the internalization mechanisms. Good question. Jayachandran, the question here is blood neutrophils, their numbers are actually different between mouse and human. So how do you think this might affect the generation and function of neutrophil-derived EVs? So now I guess we're looking at the neutrophil EVs themselves.
Yeah, I mean, I'm not sure how that would affect the actual, production of EVs from neutrophils. It might be safe to expect that you would have comparatively less neutrophil-derived EVs, just because the sheer quantity of neutrophils is lower in mouse blood, but my knowledge is very limited in that context. But it is interesting to point out that different species do have different neutrophil levels. I know non-human primate and humans tend to be more similar, but in mice, neutrophils are not necessarily the most predominant leukocyte. So it's a great point.
Good, thanks. Thank you for that. Next question is from Fred Hochberg. Hi, Fred. It's nice to see you there on the camera.
Great, Ken. Can you hear me?
Yes, we can.
So, Anne, thank you very much for your review. From a pathologic standpoint, many cancers, metastatic or primary, are filled with monocytes, histiocytes, and macrophages. Based on your review, have you been able to find any studies in which clear emergence of EVs from the tumor cell are transported to these seemingly inflammatory cells and expressed therein?
Yeah, in my brief review, I did not focus on cancerous cells and EVs and interaction with neutrophils specifically, but I do remember some titles out there involving maybe EVs derived from tumor cell lines. But yeah, my knowledge is limited there.
I mean, I'll take even a model of infection in the amoebiasis literature or parasitic literature. Were you able to find any evidence of transmission of cell-infected EVs to the seemingly inflammatory cell component?
Not that I've seen. My review was just limited to looking at basic association between cell types, but I think that would be really interesting to focus on.
So Fred, you're proposing a kind of education of these cells by EVs from a different source. Are you do you have an example of this in mind where it has been reported?
It's not been carefully looked at, and that issue has been avoided by identifying these are tumor cells and these are not tumor cells. And as a consequence, we've created a schism between what we believe are cancer cells and inflammatory cells. But it would be intriguing to use the technology that you've carefully looked at to address that particular question, whether it be in an infectious disease standpoint or a cancer standpoint.
That's a great suggestion. So, Anne, I would propose that you stick around for another year or two or 5. Maybe we can answer. There's some important questions, indeed.
Did I phrase it carefully enough, Ken, so that she stays?
That's perfect. Good. All right. Thank you, Fred. Anybody else have some questions here for Anne? All right. Well, thank you everyone for joining, and thanks so much, Anne, for taking this on, for reviewing these papers, telling us a little bit more about neutrophils, and we look forward to learning more about the ongoing experiments that you're doing, and at how this field of interaction between EVs and neutrophils develops further. So thanks again, and take care, everyone. We look forward to seeing you on a future EV Club. Bye now.